Hey, how's it going, everyone? This is FixerMed, and welcome to my high-yield biochemistry review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 2 of my multi-part series covering the entire discipline of biochemistry. So without further ado, let's go ahead and get started. So I want to open up with a very high-yield topic called glycolysis, which many of you guys should already know. Glycolysis is a vital cellular metabolic pathway that takes place in the cytoplasm, which involves the breakdown of one molecule of glucose into two molecules of pyruvate. The process consists of 10 sequential enzymatic reactions, as seen on the diagram to the left, each catalyzed by specific enzymes. In the initial energy investment phase, 2 ATP molecules are utilized to activate glucose, leading to its conversion to fructose 1,6 bisphosphate. The subsequent energy yielding phase results in the production of 4 ATP molecules, so you have a net gain of 2 ATPs, along with 2 molecules of NADH. Glycolysis plays a central role in cellular energy production, providing substrates for the citric acid cycle and serving as a crucial pathway for both energy generation and the synthesis of metabolic intermediates. So, one tip I'd say is memorize this pathway, use Anki, print the slide out, do whatever to memorize this pathway. Extremely high yield. You can screenshot this picture, print it out, screenshot it, put it in your iPad, write the pathway out with the first slide as seen here. Do whatever you had to do to memorize this pathway. It's extremely high yield. Glycolysis is involved in a lot of organ blocks, or the effects and after effects of it are. So make sure you get this down firm right now, if possible. ASAP. Moving on, let's talk about glycogenesis. Glycogenesis is the process by which cells synthesize and store glycogen, a branch polysaccharide composed of glucose units. The process begins with the uptake of glucose, which is converted to glucose 1-phosphate. Activation of glucose 1-phosphate occurs through the addition of uridine triphosphate, UTP, resulting in the formation of uridine diphosphate glucose, UDPG. Subsequently, the enzyme glycogen synthase facilitates the elongation of the glycogen chain by catalyzing the transfer of glucose from UDPG to the growing chain, forming alpha-1,4 glycosidic bonds. The introduction of branches is achieved by the branching enzyme forming alpha-1,6 glycosidic bonds and creating a highly branched glycogen structure. Glycogenesis is a tightly regulated process that occurs in response to elevated blood glucose levels, allowing cells to store excess glucose for future energy needs. I have a diagram here. As you can see, glycogenesis is the process of building up to glycogen. So from glucose, you go to glycogen. Well, the reverse is called glycogenolysis, which we'll cover now. Glycogenolysis is the catabolic process responsible for breaking down glycogen into glucose units to provide a rapid source of energy. This metabolic pathway becomes essential during periods of increased energy demand or low blood glucose levels. Glycogen phosphorylase, activated by hormones such as glucagon and epinephrine, initiate this process by cleaving glucose residues from the non-reducing ends of the glycogen chain, releasing glucose 1-phosphate. To facilitate glucose release, debranching enzyme removes terminal glucose residues from branches, converting them into glucose 6-phosphate. Subsequently, glucose 6-phosphatase, primarily active in the liver, converts glucose 6-phosphate to free glucose, which can be released into the bloodstream. Glycogenolysis ensures a quick response to energy needs by mobilizing stored glycogen reserves, particularly in tissues such as the liver and muscles. So I have the chart here. Do the same thing. You can screenshot this and then screenshot an empty copy that I managed to create. Fill it out. Know the steps, know the enzymes, write it out, use Anki, do whatever to ensure that you have these steps memorized. A lot of biochemistry is just, unfortunately, memorizing a bunch of stuff and then regurgitating it on the test. So that's my best high yield tip for this type of biochemical pathways. Moving on, let's go to everyone's favorite way of learning this, NBME application-based reasoning. So we have an early 20s male patient that consumes an energy drink. Which of the following irreversible enzymes demonstrates increased activity. You guys should know what the irreversible enzymes are from the glycolysis sheet I produced earlier. Memorize it. I'll give you guys a couple more seconds. If you don't have your answer now, go ahead and just pause the video while you think through this, or go back into video to look at the glycolysis pathway. And let's go. The answer to this question is pyruvate kinase. Caffeine stimulates the release of epinephrine, also known as adrenaline. Epinephrine activates the cyclic AMP pathway. During this, protein kinase A is activated. Protein kinase A can phosphorylate and activate protein kinase, which causes the conversion of phosphoenol pyruvate to pyruvate at an increased rate, which increases the rate of glycolysis. This causes increased glycolytic activity to contribute to the perceived boost in energy. All right, sweet. Moving on to our next NBME application-based reasoning problem. We got an early 20s male. He goes 3 weeks without food. Break down to which of the following most likely explains his ability to maintain blood glucose levels. I even gave a nice histo slide here, so if you're a histo fan, you should be able to easily pick this answer out if it's given to you on a CBSE or Step 1 question. Give you guys a few more seconds. All right, if you aren't having an answer ready yet, just go ahead and pause the video. I'm about to move on. Moving on now. Explanation. The answer for this question is skeletal muscle protein. Glucogenic amino acids will be liberated by muscle, which will then be converted to glucose by the liver. The muscle protein lacks glucose 6-phosphatase, so that does not directly produce glucose via gluconeogenesis. I hope that makes sense why it's skeletal muscle and not another type of muscle like cardiac muscle. Moving on. We have an early 20s female. She goes for a run. Which enzyme will most likely be activated initially by exercise in this patient? Give you guys a few more seconds. If you guys don't have your answer, I'm about to move on, so just go ahead and pause the video if you need more time. Moving on now. The answer for this question is phosphorylase kinase, which will phosphorylate and activate glycogen phosphorylase, the main enzyme that removes glucose 1-phosphatase units from long glycogen branches and chains. So before anything can occur, glycogen phosphorylase needs to be phosphorylated and activated by phosphorylase kinase. After the glycogen branch has enough glucose 1-phosphate units, where it's only 4 glucose 1-phosphate units in length, the first debranching enzyme, 1,4-glucosidase, removed 3 glucose 1-phosphate units, and the second debranching enzyme, 1,6-glucosidase, removes the last glucose 1-phosphate unit. Hope that makes sense on why phosphorylase kinase is the answer here. Let's move on to the next one, glycolysis enzyme information with you guys. Hexokinase is mostly ubiquitous. This means that it's found in multiple organs, basically found nearly everywhere. It decreases the Km and decreases the Vmax, and it's not upregulated by insulin. Meanwhile, glucokinase is found in the liver and beta islets of Langerhans cells in the pancreas. This increases the Km and increases the Vmax, and also is upregulated by insulin. So just wanted to give you guys a little bit of key information there. Moving on to the next slide. We have another NBME application-based reasoning question. We have a 15 to 20-year-old female with frequent thirst and urination problems, with a markedly elevated serum glucose and ketone level, alongside a serum pH of 7.1. Which of the following enzymes' activity is increased after administration of insulin? Hint: It's either hexokinase or glucokinase, so focus on those two and your reasoning behind why. And then on the NBME question bank, they probably have a bunch of other distractor answer choices, so just make sure you have that firm understanding down. All right, I'll give you guys a few more seconds. If you need some more time, just go ahead and pause the video. I'll be moving on now. So the answer to the question is glucokinase. Glucokinase is the hexokinase equivalent in the liver and is upregulated by insulin. Hexokinase is found elsewhere in the body and not upregulated by insulin. Compared to hexokinase, glucokinase has increased Km and increased Vmax. Sorry about that. I don't think I can fix that typo right now, but they do have a increased Km and increased Vmax, which means that it has less affinity for glucose and greater capacity to handle it. This makes sense since the liver should not be preferentially processing glucose over other cells in the body. Example, muscle brain. When serum glucose levels have risen high enough, the liver can act as a buffer to process glucose into glycogen for glycogenesis, as explained earlier. Let's move on to the next problem. NBME application-based reasoning. We have a 15 to 20-year-old female that eats a meal. Which of the following molecules is most likely to simulate glycolysis in this patient? All right, if you guys need more time, just pause the video. I'm going to be moving on to the next slide right now. The answer for this question is AMP. AMP and ADP simulate glycolysis, while ATP, NADH, citrate, and alanine inhibit glycolysis. Very high yield factoid to know. AMP is considered to be a more sensitive indicator of low energy levels due to its direct relationship with ATP breakdown. AMPK gets activated due to an increase in AMP to ATP ratio. AMPK activation is part of the cell's adaptive response to low energy questions. Therefore, this question specifically highlights AMP due to the cellular response to the low energy and the role of AMPK in stimulating glycolysis. So I hope that makes sense to you guys. So next NBME application-based reasoning question. We have a 60 to 70-year-old female that eats a meal. Which of the following molecules is most likely to upregulate the rate-limiting step of glucose utilization in this patient? Give you guys a few seconds to think through this. All right, I'll be moving on to the next slide now. So if you need more time, please pause the video. Moving on now. The answer for this question is fructose 2,6-bisphosphate. The molecule fructose 2,6-bisphosphate is an allosteric activator of phosphofructokinase 1, also known as PFK1, which is the rate-limiting enzyme in glycolysis. PFK1 catalyzes the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, a crucial step in glycolysis. When insulin is high, aka when you're in the fed state, some of the fructose 6-phosphate from glycolysis rather than turning into fructose 1,6-bisphosphate is shunted into a side pathway to make fructose 2,6-bisphosphate. So sorry for the typo again, but fructose 6-phosphate, instead of turning into fructose 1,6-bisphosphate, is shunted into a side pathway to make fructose 2,6-bisphosphate, basically, in this question. Let's go ahead and finish off with our final NBME application-based reasoning question and our second to last slide, so penultimate slide here. Middle-aged female, 40 to 50 years old, goes for a run. Which of the following combinations best reflects glycogen phosphorylase in the patient? Answers are combinations of phosphorylated versus dephosphorylated, active versus inactive. And I'd like to apologize. This is actually not the second to last slide. I have another problem with a similar style of reasoning, so my fault. All right, I think I gave you guys enough time. Let's go through the explanation. If you need more time, pause the video. I'm moving on now. So for the first step, are we fed or fasting? We have to find out whether we're fed or fasting. As you can see in the vignette, she is more so in the fasting state because she went for a run. Second step, would you expect a given enzyme to be active or inactive in this setting? I'd say it's pretty active. I'd say the enzyme is active. Third step, is insulin up or down? I would say the insulin levels are down because you're more so in the fasting state. And fourth step, if insulin is up, then the enzyme is dephosphorylated. If insulin is down, then the enzyme is phosphorylated. So from our findings, we can say that the answer is phosphorylated and active. Glycogen phosphorylates breaks down glycogen in the fasting state. Glycogen synthase builds up glycogen in the fed state. Insulin dephosphorylates, glucagon phosphorylates. Remember that statement. Very high yield. Moving on to the next slide. NBME application-based reasoning. We have a mid-20s female that eats turkey dinner. Which of the following combinations in terms of inactive versus active, phosphorylated versus dephosphorylated best reflects her PFK2? Give you guys a few more seconds. All right, moving on to the next slide. If you need more time, feel free to pause the video. All right. Are we fed or fasting? She just had a turkey dinner, so that seems like a pretty fed state to me. Would we expect a given enzyme to be active or inactive in this setting? I'd say the enzyme would be active. Is insulin up or down? I'd say insulin is up here because she's in her fed state. If insulin is up, then the enzyme is dephosphorylated. If insulin is down, then the enzyme is phosphorylated. So she's in the fed state, enzyme's dephosphorylated. The enzyme is also active. I hope that helps you guys. I really appreciate the support on these videos. If you have any questions, comments, or concerns, or any constructive criticism, please feel free to leave them down in the comment section below. As always, this is FixerMed signing off. Hope you guys have a great and wonderful day studying biochemistry, one of the most dry subjects in all of medicine. Enjoy, guys. Goodbye.